Hello, hello. We're going to be doing 2 lectures in one again today, but we're going to start with phylogeny and cladistics. So to start off, a little bit of background. We're going to talk about Carl Linnaeus. He is considered the father of taxonomy. Taxonomy is just the system of classifying and naming organisms. So Linnaeus specifically is why we have like scientific names for animals. But he really started this when he wrote Systema Naturae in 1735, which was a whole book of classifications, scientific classifications for animals and plants. And he classified these with a hierarchical classification, which is a system of grouping things according to their levels and orders. And before Linnaeus kind of did this, there was really no standard way of classifying any kind of organism. So names were not always even across the board. People were just kind of naming things however they felt like, which made it really confusing. And there were a lot of mix-ups. And so he kind of helped standardize this, which made it much easier for academics and really everyone to kind of get on the same page. And what really made this revolutionary is that he started using binomial nomenclature, which is where you use two terms to denote a species of living organisms. So this is where we get this idea of having a genus and a species. And of course, we have expanded that. There's families and subfamilies and orders and all kinds of other things. But he really made this kind of the norm. And with Linnaeus's version, he actually divided humans into different subspecies. So he had Homo sapiens and Homo neanderthalensis. This is kind of debated even currently. Some anthropologists argue that it is Homo sapiens and Homo neanderthalensis. Some argue that Neanderthal should actually be called Homo sapien neanderthalensis, but we'll get into that more later on in the semester. He also labeled all apes as Homo troglodytes. Of course, he only actually knew about chimps and orangutans, so it was not a great system. We do not call them that anymore. Chimps are considered pan-troglodytes, and orangutans are part of the genus Pongo. Here you can see this kind of hierarchical classification. We start with a kingdom, then a phylum, class, order, family, subfamily, genus, and then finally species. And here we have this human classification. So we are, of course, in the kingdom Animalia because we are animals. We are part of the phylum Chordata because we have a spinal cord. Part of the class Mammalia because we are mammals, the order Primates, the family Hominidae, the subfamily Homininae. Our genus is Homo. All human species are. And then our actual specific species name is Sapiens. And here, a little fun fact at the bottom. We actually share about 98% of the same DNA as gorillas and chimps. Other great apes are our closest related primate species, with chimps, which are pan-troglodytes, and bonobos, which are pan-paniscus, being our most closely related primates. So when we talk about species, we really consider species to be anything that can kind of reproduce and create viable offspring. And by viable, I mean fertile offspring. Obviously, there are species that can reproduce and have offspring. So tigers and lions can reproduce and have ligers, but ligers are not viable offspring. They're not fertile, so they can't continue to reproduce. The same thing happens if you mix a horse and a donkey. They can have a mule, but that mule is infertile. Now, there are two types of speciation. There is anagenesis, which is evolutionary transformations in one species over time. So this is going to be unilinear. There's not going to be any branching, and it doesn't result in any more species. It's just the change or the evolution of the same species. We also have cladogenesis. So this is going to be the formation of one or more new species from another over time. So it is multilinear or branching. There is a special case of cladogenesis called adaptive radiation, and this is when we see rapid diversification of a species with changing environmental conditions. So a really good example of this is during the Miocene. There's something called the ape fluorescence. Sometimes it is jokingly called the planet of the apes. Apes actually appeared about 23 million years ago. And during the Miocene, there were just really, really good environmental conditions. And it led to adaptive radiation, where there was just an incredible amount of speciation occurring. We had over 100 species of Miocene apes. For some context, there are only about 20 ape species today. So moving on, we have clades and cladistics. These are based off that cladogenesis idea. A clade is just a group of organisms that we believe have evolved from a common ancestor. And we group these animals into clades so that we can build cladistics or cladistic trees, which is really a method of classification that sorts animals and plants according to the proportion of measurable characteristics that they have in common. So a lot of times this is based off of physical attributes that you can see. Of course, now we can kind of fine tune it based off of DNA evidence, but it is assumed that the higher proportion of characteristics that two organisms share, the more recently they diverged from a common ancestor. So that's how we get into more specifics within our kind of hierarchical classification. So obviously all mammals, they give live birth, they produce milk, that kind of stuff. So we can assume that at some point in time, way back when, any animal that currently does that was coming from a common ancestor. Now, this isn't always the case because you can have traits that are similar, like wings in bats and birds, but they do not have a super close common ancestor. And here we'll kind of talk more about that. So these characteristics that animals can have or develop, these evolutionary characteristics, they can be primitive or ancestral. These are going to be features shared by a group of species by virtue of being present in a remote common ancestor. So for example, eye orbit. All primates have enclosed eye orbitals, and that is considered a primitive feature or primitive characteristic because it is shared with a common ancestor. It's something that we have all passed down from that common ancestor. They can also be derived, and these are going to be features that are shared uniquely between two or more descendant species. So, for example, large brains. These are basically going to be more recently evolved characteristics that less species in a clade share. So, for example, humans and Neanderthals have relatively larger brains than other primates compared to our body sizes. And that would be considered a derived feature rather than a primitive because it isn't something that we share with a common ancestor. And it is important to note that what is considered a primitive trait versus a derived trait is going to be dependent on what you are looking at specifically in your cladogram. And I'll show you a little bit more about that in a couple of slides. Along with homology, you can also have homoplasy, and these are going to be character traits shared by species that isn't present in a common ancestor. So this is going to be something that has resulted as independent evolution or parallel convergent evolution, whatever you want to call it. These are going to be like our bat wings versus our bird wings. They are both wings, but they don't share a common ancestor that was a primitive trait for. They just happen to both evolve to have that trait because it helped them adapt to their environment. Here we can see an example of cladograms. This is not what your guys' cladogram should look like. This just is kind of a helpful diagram to help understand how traits are assigned. So we always start with our older ancestors. So these are going to be whatever species or organism has more of the primitive traits, doesn't really have any special derived traits. It's going to be very basic. Typically, this is also going to be your older species. And then we're going to branch off. And you can see we have this little diamond here. We have an upside down triangle, and we have this little asterisk or star. These are all going to represent traits. So our Y is going to be our kind of first ancestor in this. And then we're going to branch off. We get to species X, and it's developed this, what would be a derived trait for the species. We're also going to develop C, which is going to have this upside down triangle. That would also be a derived characteristic for that species. But once we move on to species A and B, we see that they share, and we're looking at the cladogram on the left-hand side, by the way. This A and B are going to share this diamond. That is now a primitive trait for them because they are developing it from species X, so from a recent common ancestor. However, this upside down triangle is not shared or derived is a bad word to use. It isn't shared with species C as a common ancestor. So this would be an example of your homoplasy. Meanwhile, species A is going to develop this little asterisk. This is going to be a new derived trait. When we look at the diagram 2 on the right-hand side, you can see that we have something very similar going on, except for this time B and C do share a common ancestor, and so that upside down triangle, whatever trait that is representing, is actually going to be the primitive trait instead of the derived trait in this example. Here's a little bit easier to see. You will see this exact same cladogram on your homework as well. We're starting out with some common ancestor. We don't know what it is right now. And this is what your cladogram should look like. You will have all of your species in a row at the very top, and then you will have them branching down. We don't know what the common ancestor is here, but that's okay. We just know that there is one, and whatever common ancestor that all of these species share had the primitive traits of Catarrhine noses and a 2-1-2-3 dental formula. Then you can see we branch off into Papio. This is going to be baboons, by the way. And they're going to share these primitive traits. They're also, with Pan, going to share large brains, misspoke. They're just going to share those primitive traits. Then we can see here that we have large brains and highly stable wrist joints right before Pan or chimps branch off. And you're going to see a branch every time we have new characteristics or traits they're going to show where these differ. Sorry, I've got an ant bugging me. Then we move up and we get to Homo sapiens, Homo neanderthalensis, and Australopithecus. And you can see before we branch off with Australopithecus and then our Homo species, we have another little trait label that talks about bipedalism, reduced canine dimorphism. And then when we move up to Homo sapiens and Homo neanderthalensis, where they branch off, we see relatively large brains and symbolic behavior. So when you're reading this, whatever is above the traits is going to share those. So they all are going to have Catarrhine noses and this 2, 1, 2, 3 dental formula as a primitive trait. But Papio is not going to have large brains, highly stable wrist joints, bipedalism, reduced canine dimorphism, or symbolic behavior because it is below where those traits split off for other species. Likewise, Australopithecus is not going to have the relatively large brains and the symbolic behavior that Homo sapiens and Homo neanderthalensis do. But it is going to share bipedalism, reduced canine dimorphism, large brains, highly stable wrist joints, Catarrhine noses, and the 2, 1, 2, 3 dental formula. So if we are ignoring Papio and Pan and we're just looking at our Homo species and then Australopithecus, we could say that large brains and highly stable wrist joints are actually a primitive trait for them, whereas relatively large brains and symbolic behavior are a derived trait for the Homo species. However, if we are looking at the Papio and the Pan, then the primitive traits are going to be the Catarrhine noses and the 2, 1, 2, 3 dental formula, and Pan is going to have derived traits of large brains and the highly stable wrist joint. It won't, however, have the bipedalism, reduced canine dimorphism, or the symbolic behavior that we see higher up in the cladogram. You will also see this on your homework because you guys are going to create your own cladogram. This is a very common way to kind of map out the traits that are going to be important for a cladogram. You can see here that we have our features. We have a nose, tail, the os centrale. This is your wrist bone. And then thumbs. And we have species A through E. So when you do your cladogram, those should be written across the top in just an even line. And we can see for the nose that all of them have a drooping nose except for species E. For the tail, all of them have no tail except for species D and E. The only two that have a fused os centrale is the species A and B. And then they all have opposable thumbs. So through this, you can tell that the opposable thumbs is going to be a primitive trait because all of the species share it. Then you're just going to move up into what is more and what is less shared. So things that are less shared are going to be higher up in your kind of branches or your tree. And things that share more are going to be lower. So we would have, let's see, thumbs is going to be your most primitive trait. Noses are also going to be relatively low on your cladogram because more species share it. And then your tail and your os centrale are going to be up further in the top. We know that species D and species E need to be lower in your cladogram because they are sharing more of these traits that species A, B, and C do not share with them. And if you guys need help with this, you can, of course, let me know. If you want me to check your cladograms before you turn them in, then you can definitely email me those. I do prefer if you draw them rather than trying to do them, you know, like typing or any other way, because it doesn't always show up very well on Blackboard. And whenever you're doing a cladistic, there are a couple of assumptions that you need to keep in mind. So you need to be able to list the general characteristics that are important to the species. This can be in this box that we saw before. For each characteristics, you need to record the different state in which it appears. Then you need to rank the character states as primitive or derived and compare the frequencies of each. So which ones have more primitive traits versus more derived traits. And then you need to sort these species into clades using the derived characters. So the fewer or the more derived characteristics that you have, the higher up in the cladogram it should be. You should also watch this video and kind of think about these questions.